Hi everyone, and welcome to the BME2104 lecture on enzymes. This is an amazingly diverse family of proteins that catalyzes pretty much all the chemical reactions in our bodies. Today we'll talk about how enzymes speed up chemical reactions by stabilizing their transition state and lowering the activation energy of the reaction. We'll also learn how to model enzyme behavior mathematically with Michaelis-Menten kinetics, and we'll learn how to use the Hill coefficient to describe enzyme cooperativity. Finally, we'll talk about how enzyme activity is turned on and off inside the cell by allosteric regulation and by different types of inhibition. And the human disease topic that I'll use to illustrate key aspects of enzymes and their inhibition is viral polyproteins. Specifically, we'll talk about an enzyme activity related to polyproteins that's required for the SARS-CoV-2 viral life cycle. And then we'll talk about how this enzyme is being targeted for inhibition by the pharmaceutical industry as a potential treatment for COVID-19. So let's start out by talking about polyproteins. I'll give a definition for polyproteins in a second, but first I want to explain that viral genomes are incredibly compact because all the DNA or RNA in the viral genome has to fit inside of the virion particle. The diameter of the SARS-CoV-2 virion is approximately 100 nanometers, the same width as a single stack in the Golgi or ER, and the entire viral genome has to be packed inside of there. This places an incredible constraint on viral genome size. As you can see here, the SARS-CoV-2 genome is approximately 100,000 times smaller than ours, because the virion structure just can't physically accommodate anything larger than around 30,000 nucleotides in length. In order to save space and use their genome as efficiently as possible, many viral proteins are expressed as polyproteins and then cleaved by protease enzymes. Let's explain what this means. In mammalian organisms like ourselves, the genes on our chromosomes are well separated and spread out, and each gene codes for a single protein. In viral genomes, we have the coding region for multiple proteins all compacted together, and this is translated as a single long polyprotein that has all its individual protein components linked together from N-terminus to C-terminus on a single polypeptide chain. Processing this viral polyprotein into its individual protein components is a key step in viral maturation, and this job is accomplished by a specialized enzyme called a protease, which chops up the polyprotein into its individual functional protein components. This protease enzymatic activity is required for the viral life cycle, and we'll talk at the end of lecture today about efforts to design drugs that inhibit this activity as a potential anti-COVID-19 therapy. Alright, so up to this point, we've talked a lot in this class about amino acids and proteins, but we haven't talked very much about what proteins can do. One thing that proteins can do is act as enzymes, so let's define what enzymes are. Enzymes are biological catalysts. A catalyst is something that accelerates the rate of a chemical reaction. One thing that's important to remember is that catalysts don't change a chemical equilibrium, they just help that equilibrium be reached faster. That said, we typically think of enzymes as acting on substrates to yield products, where substrates are the reactants on the left-hand side of the reaction, and the products are the chemical compounds on the right-hand side. As an example, I show here the chemical reaction that converts hydrogen peroxide into water and oxygen. This reaction is catalyzed by an enzyme called catalase. As a side note, catalase uses iron as a cofactor, kind of like what we talked about last week, how hemoglobin uses the cofactor heme to bind oxygen. Some enzymes catalyze the reaction of small molecules like hydrogen peroxide shown here, but many other enzymes in our body catalyze the reaction and chemical modification of larger biomolecule reactants, such as proteins, nucleotides, and lipids. So what are the qualities of enzymes? Well, first of all, they're efficient. Enzymes can take a reaction that is energetically favorable but kinetically slow, something that would take thousands of years to complete, and they can catalyze the same reaction to occur within minutes or even seconds. Enzymes are also specific. They will often recognize specific substrates but not closely related molecules, not even stereoisomers. We didn't talk about stereoisomers during our amino acids lecture, but many of you will be familiar with this concept from introductory chemistry. The alpha carbon in every amino acid is a stereocenter because it has 4 unique constituents bound to it: the amine, the carboxylic acid, the side chain, and a hydrogen atom. And therefore, every amino acid has the potential for 2 different 3D orientations with mirror symmetry. Those are stereoisomers. All naturally occurring amino acids exist as the L isomer, and enzymes that act on proteins will only react with L amino acids, not with the D isomer amino acids. Another property of enzymes is that they're saturable. I'll illustrate this point on the right-hand side. In this plot, the rate of chemical reaction is on the y-axis, and the concentration of substrates is on the x-axis. As you can see in this example, the rate of reaction increases with increasing substrate concentration, but when you get up to a certain concentration of substrate, the reaction rate levels off in a plateau. This is because the enzyme is saturated. It can't bind reactants and convert them to products any faster. Enzymes are also tightly regulated. Based upon the state of the enzyme, whether it's bound to an activating regulatory protein or whether it's been covalently modified, these factors can affect the rate of catalysis. One more example of this type of enzyme regulation is inhibition. This reduces an enzyme's rate of catalysis. If we add an inhibitor to the reaction on the right, it might look something like this. We'll talk about different types of enzyme inhibition at the end of lecture today. There are many different types of enzymes in the cell. The general naming convention we follow is to name proteins that act as enzymes with the suffix ase. So, if you see a protein name that ends in ase, then you know that protein is an enzyme. In mammalian cells, we often have different versions of an enzyme that catalyze the same reaction. These are called isoenzymes, or just isozymes. It may seem redundant, but having different versions of an enzyme that catalyze the same reaction, but are localized or regulated differently, can help cells respond appropriately to a wide variety of environments and situations. Let's go over a couple general classes of enzymes now. Proteases and peptidases catalyze the hydrolytic cleavage of peptide bonds. Proteases cleave proteins into peptides, and peptidases cleave peptides into amino acids. We'll talk about SARS-CoV-2 proteases at the end of lecture today. Also, in our first lecture, I mentioned acid hydrolases in the lysosome. Acid hydrolase is a large family of protease enzymes which hydrolyze their substrates in an acidic environment. All proteases use hydrolytic cleavage with a water molecule to cleave the covalent peptide bond. This is the opposite of the condensation reaction by which the bond was formed in the first place. Another class of enzymes is called kinases. These are very important for cell signaling, and they act by adding phosphate groups to different biomolecules. One especially important type of kinase is protein kinases, which phosphorylate other proteins. In our lecture last week on proteins and amino acids, we went over the residues that are phosphorylated in mammalian cells: serine, threonine, and tyrosine. When these residues on a protein are phosphorylated, this leads to a significant change in their charge and steric bulk, which will change the behavior of the phosphorylated protein. This is how many enzymes are turned on and off. So you have this one class of enzyme, protein kinases, regulating many other enzymes in the cell. Protein phosphorylation is also the most common and best studied mechanism for how cell signaling pathways are propagated throughout the cell. There are also enzymes that catalyze the breakdown of other biomolecules in the cell. One example of these is phospholipases, which cleave phospholipids in cell membranes. There are many other types of enzymes. Synthases synthesize things, ligases ligate things together, and there's many, many more. I won't ask you to memorize every class of enzyme, but you should be aware that there are many other types, and they're usually named in a logical way so you can figure out what their function is from their name. Alright, so how do enzymes perform catalysis? One thing they must do is bind their substrate in a reversible manner and provide an environment that accelerates the chemical reaction by lowering its activation energy, as we'll discuss in a few slides. The site where an enzyme binds its substrate is called the active site, also known as the catalytic site. If you look at the structure of an enzyme binding its substrate, it will almost always appear that the active site is shaped to perfectly fit its substrate, along with having complementary side chain residues that favor substrate binding interactions, such as favorable hydrophobic and electrostatic interactions. In rare cases, this shape is present when the substrate is not bound, making it like a lock and key, where the substrate is a perfect fit. But what's far more common is to see that the enzyme active site changes and molds to fit the substrate upon substrate binding, which is more like an induced fit. Instead of a perfect fit of two complementary puzzle pieces like the lock and key model, there's really more of a back and forth. The protein conformation changes to accommodate the substrate, and substrate conformation is reoriented as well. This is called the induced fit model and is a more accurate way to think about an enzyme binding its substrate. Here on the right, I'm showing botulinum toxin that I used to use as a teaching example for this lecture. You can see in this structure that it's bound to its substrate, which is shown here with space-filling spheres. This is a good example of induced fit binding. You can see how the active site is wrapped around the substrate. Let's look at another example to see this all a bit more zoomed in. What I'm showing here is the active site of the protein PI3K and how the structure of its active site changes depending on which small molecule inhibitor it's bound to. This comes from the first scientific paper I was ever an author on, way back in 2006. I put a link to the paper in the lecture notes in case anyone wants to read more about this. Alright, so in addition to the active sites on the previous page, I'm showing a few more here. What I've done is pull up the protein structures of well-known examples of the enzyme categories I introduced 2 slides ago. Thrombin is a protease important for blood coagulation. Epidermal growth factor receptor, also known as EGFR, is a kinase that plays an important role in many cancers. Phospholipase A1 is a lipase that acts as a digestive enzyme for phospholipids. We discussed these in our previous lecture on the cell membrane. In these pictures, the surface of the enzyme is colored gray, and what I'm highlighting in red are the residues that directly participate in enzyme catalysis. As you can see, in the vicinity of these catalytic residues is a groove or pocket. This is the active site where the substrate is bound and oriented with a correct geometry that allows for the catalytic residues to catalyze the reaction. The next thing I want to talk about is how enzymes accelerate the rate of a chemical reaction through the process of catalysis. How does this work mechanistically? To talk about this, we need to introduce the Gibbs free energy, also known as G, which is a combination of entropy and enthalpy for the system. The change in free energy, or delta G, determines whether a reaction is favorable or unfavorable. Any process with a net negative delta G is thermodynamically favorable. We can use the Gibbs free energy to track the progress of a reaction from substrates to products, as shown on the right. On the y-axis, we have free energy, and the x-axis shows the course of the reaction, starting with substrates and going to products. Altogether, this plot shows us how the free energy changes over the course of the reaction. The difference between the free energy of the substrates and the products is the delta G for this reaction. As long as delta G is negative, this reaction is thermodynamically favorable and will occur spontaneously. An important question, though, is how long the reaction will take to proceed. The rate of reaction is determined by the height of this hump in the middle, and the height of this hump is called the activation energy. This is the barrier the reaction must overcome to proceed, and the higher the barrier, the longer it's going to take random thermal fluctuations to push it over that activation energy. We always see this higher energy bump in reactions like this. There's always an unfavorable intermediate in between the relatively stabilized reactants and products. This intermediate state is referred to as the transition state. Why is the transition state higher energy? Every reaction is different, but there are usually both enthalpic and entropic reasons why the transition state is higher energy. From the enthalpic perspective, the transition state occurs after reactant bonds are broken, but before product bonds are made. That's the enthalpic contribution. From an entropic perspective, you can think of the transition state as being held in a highly fixed position. Since there's only one possible state for this, that's very unfavorable entropically. Since the transition state is energetically unfavorable, this explains why the activation energy controls the rate of catalysis. If the transition state is highly unfavorable, then the activation energy will be high, and the reaction will proceed very slowly. The way enzymes work to speed up reactions is by lowering the activation energy. This is done by providing an active site environment that makes the transition state more energetically favorable, usually by providing polar and hydrophobic binding interactions. Let's look at another example to test your understanding of these kinds of plots, which are called reaction coordinate diagrams. If we look at the two reactions, A and B, which do you think will go faster, and why? Take a second to think about this, and then we can talk about the answer during our class discussions on Thursday. Alright, so there are a number of conditions that affect enzyme activity, which, as we just discussed, is the enzyme's ability to lower the free energy of the transition state and speed up the reaction. One such condition is temperature. In general, higher temperatures will increase reaction rates by providing additional thermal energy to push reactions over the transition state barrier. But if the temperature rises too high, the enzyme itself will become denatured and no longer able to function as a catalyst. Thermal denaturation means the enzyme begins to unfold and loses its conformational structure at higher temperatures due to excess thermal fluctuations. Another thing that affects enzyme activity is pH. This is primarily due to changes in side chain residue charge, as controlled by their pKa's. Many catalytic residues are charged, and therefore can be directly affected by changes in pH. Additionally, changes in pH can disrupt electrostatic interactions that hold the protein in its larger three-dimensional tertiary or quaternary structure, leading to denaturation and loss of catalysis, just like we discussed with elevated temperatures. Enzyme cofactors improve enzyme activity by participating in complexes that stabilize substrate binding and the transition state. Divalent metal cations are a commonly seen enzyme cofactor. Some examples include the zinc cation in tetanus toxin and the magnesium cation, which coordinates the phosphate group of ATP in the kinase active site. Another class of molecule that improves enzyme activity is coenzymes. Unlike cofactors, which just serve to get the substrates in the right place and orientation, coenzymes participate directly in the reaction chemistry by transfer of electrons or binding to either the reactants or the products. Most of the vitamins that our body needs are actually coenzymes, such as biotin, also known as vitamin H. When we get to the lectures on metabolism, we'll see a number of coenzymes that help transfer electrons between metabolites to extract energy from them. A generic example of this is shown at the bottom. The rate of enzyme activity can be described as a function of substrate concentration. This is referred to as the Michaelis-Menten equation, shown here on the bottom left. Before we go any further, I want to quickly point out that this equation should be colored blue because this is something you'll be responsible for learning. We'll use it a lot in this class. In the Michaelis-Menten equation, V, or velocity, is the rate of reaction, how much product is produced per unit time. The behavior of this equation is shown on the right. It's a hyperbolic function that approaches Vmax, the maximum rate of enzyme catalysis, as substrate concentration increases. We talked earlier about how enzymes are saturable. Well, Vmax is the asymptote here beyond which the enzyme cannot go any faster. The other term we need to introduce in this equation is the Michaelis constant Km. This is the substrate concentration at which we observe exactly 1/2 Vmax, that is, 1/2 the maximum reaction rate. You can see this illustrated in the plot on the right with green dotted lines. We can observe these behaviors simply by plugging in values to the Michaelis-Menten equation, and I'll run through a couple examples here. If we choose a substrate concentration that is much, much greater than Km, the Michaelis-Menten equation will essentially reduce to V equals Vmax, satisfying the definition for Vmax as the highest possible reaction rate that we can see at saturating substrate concentrations. If we make the substrate concentration exactly equal to Km, then the Michaelis-Menten equation reduces to V equals 1/2 Vmax, satisfying the definition for the Michaelis constant Km. As you can see here, if we want to relate an enzyme's reaction rate to substrate concentration, we only need to know two parameters for that enzyme, its Vmax and its Km. A quick note about this, however, is that Vmax depends on enzyme concentration. If you double the enzyme concentration, then you'll double the rate of reaction. For this reason, you'll often see Vmax given in units of moles product per second per mole of enzyme. All right, let's do one more test of your understanding so far. If we did an enzyme rate experiment and got the following values for velocity V at different concentrations of the substrate S, what does this make the Km for this enzyme? You should be able to figure this out with all the information given here. So take a little time to work on this, and then we can talk about the answer and how we got there during our class discussions on Thursday. All right, and now we're back from modeling the Km. What I want to emphasize on this slide is that the Michaelis-Menten equation is a model of enzyme kinetics, and there are some specific assumptions that go into this model that it's important for us to understand. Let's start with a full model of enzyme catalysis in the top left, and its associated differential rate equations. Here we're modeling the simplest type of enzyme catalysis, where a single substrate is converted into a single product. These equations can get much more complex with cleavage or ligation reactions, or if we add in inhibitors on top of the substrates. But for now, this is a good system to describe how we derive the Michaelis-Menten equation. The reaction we're dealing with works like this. We start with enzyme E plus substrate S. These are in the top left, and these have associated K-on and K-off rates for enzyme substrate binding, indicated by K1 and K-1 here. The enzyme substrate complex ES is in equilibrium with the unbound E and S species, but we also have an alternative path. The catalytic rate Kcat describes how quickly the enzyme substrate complex reacts to create product P, while simultaneously regenerating free enzyme E. Using this multi-step chemical reaction, we can write out differential equations that describe the changes in concentration per unit time for the enzyme E, substrate S, the enzyme substrate complex ES, and the product P. As a quick note, the change in product formation dP dt is the reaction velocity V that we care about, and we want to define this as a function of substrate concentration. We're not going to require you to derive the Michaelis-Menten equation for this class, but I do want to walk you through the concepts of their derivation and the key assumptions that we use to get there. The first assumption we make is that the reaction is occurring at steady state, which means that the concentration of enzyme substrate complex remains constant, not changing over time. This allows us to set the DES dt rate equation equal to 0, leaving us with a simple algebraic equation that relates the concentrations of the enzyme, substrate, and the enzyme substrate complex. The second key assumption we make is that the initial concentration of substrate at time 0 is much, much larger than the initial concentration of enzyme, so that as the reaction proceeds, the concentration of substrate doesn't change significantly. We can then combine these assumptions with the fact that concentration of free enzyme E plus the concentration of enzyme substrate complex ES has to be equal to the initial concentration of enzyme. And this gives us the equation you see underneath, where the formation of product per unit time, dP dt, is equal to Kcat times the initial concentrations of enzyme and substrate, divided by Kcat plus K-1 divided by K1, plus the initial concentration of substrate. Next, we simplify to the Michaelis-Menten equation by defining two new lumped together parameters. Vmax equals the catalytic rate Kcat times the initial concentration of enzyme, and Km equals Kcat plus the substrate off rate K-1 divided by the substrate on rate K1. Okay, so that's a quick explanation of how we derive the Michaelis-Menten equation, and it's important for you to understand the key assumptions that go into it. The reason for that is because there'll be examples in the cell where these assumptions about steady state and a limitless pool of substrate don't hold, and you'll need to know if the system you're looking at can or cannot be described accurately with Michaelis-Menten kinetics. Okay, so enough theory. How do we actually measure all of this? One of the things I mentioned earlier is that enzymes are specific, but often we can identify a synthetic substrate that's similar enough for the enzyme to accommodate it. And if we're lucky, we can identify a synthetic substrate that changes its color or fluorescent properties after it's reacted and turned into a product. As a side note, if we can't find a colorimetric or fluorescent substrate, then we can always read out and follow a reaction by other analytical methods like NMR or mass spec. There's a lot of other assays we can use, but these are usually kind of a pain and make it harder to collect lots of measurements that we'll need for doing the kinds of calculations that we're going to talk about. So we always try to find a substrate that we can read out by colorimetric or fluorescent assays if possible. Okay, So in the case of colorimetric substrates that change to a specifically colored product, we can shine a light on the reaction and measure how much light is absorbed with a spectrophotometer. We can monitor the increase in absorbance at the product's wavelength over time, and this will tell us how quickly the product is being produced. To do this, we take advantage of Beer's law that the absorbance A of any compound is a function of its extinction coefficient epsilon multiplied by its concentration C and the path length L. That's how long of a distance the light travels through solution. The nice thing about Beer's law is that absorbance varies linearly with concentration, so we can use this directly to read out the formation of product over time. As you can see, the way these reactions are usually set up, there's an initial linear phase of the reaction, followed by a slow trailing off. The reaction trails off here because the substrate is being depleted, and we're no longer satisfying the steady-state assumption of Michaelis-Menten kinetics. Therefore, when we do in vitro reactions like this to determine enzyme rate constants, we always use the initial linear regime to calculate dp dt. This is critical if we want to apply the Michaelis-Menten equations. Everything I did on the previous slide was enough to determine just one point on the Michaelis-Menten curve. We derived a single velocity dp dt for a single concentration of substrate. What we can easily do, however, is repeat the same experiment with different initial substrate concentrations, including a blank B, and then we can use a spectrophotometer to monitor the formation of product over time for each substrate concentration. With this data, we can quickly plot our Michaelis-Menten curve and then derive Vmax from the height of the asymptote and Km from the half maximal velocity. I want to mention one thing here, because students always get confused by this. The plot we're looking at here is not the same as the absorbance versus time plot on the previous slide, even though they look very similar. To repeat for emphasis, each point on this plot was derived from a single dp dt slope fitting from the earlier linear region of the absorbance plots on the previous slide. That's a key point that lots of students get hung up on, so if you don't think you're understanding this, make sure to bring this up during our small group discussions on Thursday, during office hours, or ask a question about it on Piazza. One other thing to keep in mind about enzymes: the standard way to report the quantity of an enzyme is in units rather than grams or moles. This is the convention, meaning the standard way that's agreed upon by everyone in the field. The thing that's annoying about units is that the definition varies from one enzyme to the next, depending on each enzyme's optimal reaction conditions. It can be frustrating having this non-standard unit of measurement, but it's really born out of practicality. Enzymes are relatively fragile proteins that are highly sensitive to temperature, pH, and substrate concentration, like we discussed earlier in the lecture. When we're working with any enzyme, we care more about how much activity we have rather than how much enzyme we have. This is because it's almost impossible to prepare enzyme preps with perfect consistency, but it's easy to quantify their catalytic activity and standardize based on this. I'm showing here the definition for a unit of creatine kinase activity. This comes from the company Sigma, which sells it in purified form. This is a kinase, one of the types of enzymes I talked about earlier in the lecture. And as you can see, one unit of activity is defined as the amount of enzyme necessary to catalyze the conversion of 1 micromole of creatine to creatine phosphate per minute at 25 degrees Celsius at pH 8.9. So if you buy this from Sigma at 10 units per mil, you won't know exactly how much creatine kinase protein you're getting, but you will know exactly how much you need to add to a reaction in order to see the desired result. Another way to think about enzyme activity is the turnover rate. This is the rate of the enzyme at saturation for a single enzyme molecule. So if you had a single enzyme molecule with saturating substrate, how many reactions will it catalyze per second? I'll work through an example here. The Vmax for tetanus toxin, which acts as an enzyme, is determined to be 111 picomoles per second per microgram enzyme. If we have the Vmax and we want to know the turnover rate, all we need is the molecular weight of the enzyme, and we can look this up to see that the molecular weight of tetanus toxin is 50 kilodaltons, or kDa. The term kDa is what we use for protein molecular weights. It's essentially the same as grams per mole. With this information, it's a simple calculation to determine that the turnover rate for a single molecule of the tetanus toxin enzyme is 5.5 reactions per second. I'll walk through the calculations here. And please note that these scales that I just put up, they're in blue. Not all of you will be familiar with them, but they'll be important for the calculations that we do in class, so you'll need to learn them. Going from largest to smallest, I'll start with microgram, which is 10 to the negative 6 grams, and then nanogram, which is 10 to the negative 9 grams. Many of you have seen these two before, but you might not be familiar with the next two. Picogram, that's 10 to the negative 12 grams, and femtogram is 10 to the negative 15 grams. We don't need to go lower than this. I focus on micro to femto because these are the units that usually come up in our cell and molecular biology calculations. Okay, so now that we've covered those scales, the calculation is relatively straightforward. We start out with a Vmax, which is 111 picomoles per second per microgram enzyme. If we want to convert this to the turnover rate for a single enzyme molecule, we can start by converting picomoles to moles, and then micrograms to grams, and finally grams to moles. After canceling out all the units, we're left with a turnover rate of 5.5 reactions per second per enzyme. So what this means is that when the enzyme is working at maximum velocity, one molecule of tetanus toxin is churning out 5.5 reactions per second. Alright, so we just spent a lot of time talking about how enzyme catalysis works. For the rest of lecture now, we're going to talk about how it's regulated. We'll start out with allosteric regulation, which is a general term for action at a distance in protein structures. What this means is some kind of molecular change at a position distant from the active site, which is propagated through the structure of the protein to cause a conformational change at the active site that affects catalysis. We'll see some examples where allosteric regulation turns on enzyme activity, and other examples where it turns it off, depending on which enzyme we're looking at. Remember, there's a huge variety in the structure, function, and regulatory mechanisms for all the different enzymes in our body. Allosteric regulation can modulate their activity by changing the rate of catalysis, changing the strength of substrate binding, or changing both of these. We'll see this general concept of allosteric regulation contributing to these other more specific forms of regulation as well. Covalent modification can modify enzyme activity by adding or removing molecular groups. One of the best studied examples of this is phosphorylation by protein kinases. We'll also see another type of modification, protease cleavage. This is often used by the cell to turn off an enzyme activity by chopping it up into pieces. But we also see this very interesting type of regulation where protease cleavage is required for enzyme maturation to be converted from the inactive proenzyme to the active enzyme. We also call this type of inactive proenzyme a zymogen. Another important form of enzyme regulation is the association and dissociation of subunits in an enzyme complex. Sometimes, all the subunits come together and participate in catalysis, so they're all required. In other examples, we have subunits that don't participate directly in catalysis, but can turn enzyme activity on or off by allosteric mechanisms. The last type of enzyme regulation we'll go over is this concept of cooperativity. This is where the formation of the enzyme product promotes more product to be formed, and if we see this type of behavior, it can imply that there are multiple catalytic subunits working together. Okay, so let's go into a little more detail about allosteric regulation. This is often induced by regulatory molecules that bind at a site distant from the active site, which triggers a change in the protein conformation that's propagated all the way through the protein and changes the geometry of the amino acids in the active site. In many cases, this will happen in separate domains of the enzyme. The allosteric regulator will bind to a regulatory domain, and this participates in allosteric regulation of the catalytic domain, like you see in the diagram on the bottom right. In this picture, the allosteric regulator is an inhibitor, but we'll see other examples where the binding molecule can act as an activator instead. I just introduced the term protein domain, so let's give a formal definition for this now. Protein domains are structurally and functionally distinct regions of a single protein. Each protein domain is composed of a single stretch of amino acids within the longer polypeptide chain of the full protein. If you do a thought experiment and cut a protein up into its individual domains, each domain would be able to fold properly and perform its individual function. That's part of the definition, that domains can act like standalone units. It's important to remember, though, that they're still part of a single polypeptide chain in a single protein. On the other hand, enzyme subunits come from separate proteins each coded by a separate gene, and these bind together to form multi-subunit enzyme complexes. One really neat example of an enzyme with different protein domains is the botulinum toxin. This is the molecule that causes toxicity in botulism poisoning, and it's unusual because it's actually an enzyme itself. In many ways, it's a protein that acts like the viruses we've been learning about so far in this semester. It has a receptor binding domain that binds to receptor proteins on the cell surface of motor neurons and facilitates internalization by endocytosis. Then, it has a translocation domain that effectively acts like a spear to punch a hole in the endosome membrane. This happens when the endosome pH becomes more acidic, and it becomes more acidic as it's being converted into a lysosome, and this causes a conformational change in the translocation domain that punches through the endosome membrane. And then once the protein is inside the cytoplasm, the protease inside the cytosol, I should say to be more precise, once it's in the cytosol, the protease activity of the catalytic domain targets a specific protein that's required for neurotransmission. This is how botulism poisoning leads to paralysis. It's a really amazing feat of evolution, putting all of this together into a single protein. And there's even more cool details that I'm not showing here. So if any of you want to learn more about this, I'd be happy to share more of my notes from last semester. Alright, so let's move on now to different mechanisms for regulation of enzyme activity by covalent modification. The first one we'll talk about is protein phosphorylation. Here's a general scheme for how this can work. A regulatory protein kinase transfers a phosphate group onto an enzyme, and this causes some kind of allosteric change that activates enzyme activity. As a reminder, the amino acids that can get phosphorylated on mammalian proteins are serine, threonine, and tyrosine, all the amino acids that have OH hydroxyl groups on their side chain. One place we see this type of regulation a lot is actually in the activation of protein kinases themselves. Later in the semester, we'll talk about kinase signaling pathways, how one kinase activates another kinase, which activates another kinase to propagate some kind of signal throughout the cell. One mechanism that's often used to activate kinase enzyme activity is amino acid phosphorylation on a special region of the kinase protein called the activation loop. When this is phosphorylated, it causes an allosteric change that brings the kinase into its active conformation. This is illustrated in the molecular animation of the kinase FGFR1 here. After phosphorylation of two tyrosine residues on the activation loop, it swings around like this, and this has two consequences. First, it tweaks the active site to allow ATP binding. Remember that ATP is the phosphodonor for kinases. Second, the new conformation of the activation loop forms a binding platform for the protein substrates of this kinase. So as you can see, activation loop phosphorylation is a really important mechanism for regulating the activity of protein kinases. Let's just watch this go around one more time. So here we have the active conformation, and then when they're dephosphorylated, it swings back into the inactive conformation. Protein phosphorylation can regulate enzyme activity by other mechanisms in addition to the direct allosteric changes we discussed on the previous slide. One of the most common mechanisms is by changing the enzyme's localization. This doesn't directly change activity at the enzyme active site, but it has a huge practical effect on the rate of enzyme catalysis due to substrate availability. We see this with many enzymes. They're always quote-unquote on, but they need proper localization to bring them into contact with their substrates. After phosphorylation, some proteins may be localized to the plasma membrane by specific binding domains that recognize and bind tightly to their phosphorylated form. This same mechanism can target enzymes to the nucleus or other compartments in the cell, depending on the location of their binding partner. Another way that protein phosphorylation can regulate enzyme activity is by recruiting different binding partners. This is very similar to what we just talked about for localization, and often there's overlap between the two. An enzyme will be localized somewhere, and its active site catalysis will be modulated allosterically by its binding partner. So far, I've only given examples where protein phosphorylation activates enzyme activity, with phosphorylation that recruits an activating binding partner. I want to emphasize here that we'll often see the reverse as well. Depending on the protein, its phosphorylation may recruit an inhibitory binding partner instead that binds and inactivates the enzyme. We see this kind of inactivation by phosphorylation in other modes of regulation that we already discussed too. Localization can be used to sequester away from substrates rather than provide access to them. and the type of induced allosteric changes we discussed on the previous slide can turn an enzyme off instead of turning it on. Okay, one last type of covalent modification I want to go over in more detail is protease cleavage. As I mentioned on a previous slide, this can activate proenzymes by converting them into the active enzyme form. So let's go into more detail about that here. Proenzymes will often have inhibitory prodomains that prevent enzyme catalysis by physically occluding the active site. As you can see in this animation, all we need to do to activate the enzyme is cleave off the inhibitory prodomain, which opens up the active site, and now the enzyme is ready to go. One thing I want to point out here is that this is an irreversible change. Once the prodomain is cleaved off, it diffuses away and it's never going to be religated back on. This is different from protein phosphorylation that we were discussing earlier, because phosphate groups can be taken on and off, so the enzyme can be continuously regulated in this way. Kinases can be turned on and off, and then on and off again. We'll talk more about how that happens later in the semester during our lecture on cell signaling. Alright, now let's move on to talk about enzyme regulation cooperativity. This concept is a little tricky, so I want to spend some time on it. In enzyme regulation, cooperativity refers to a scenario when product formation promotes more product formation. This can imply multiple catalytic subunits, or it can imply other types of self-reinforcing catalysis. It's similar to positive feedback in how it's self-reinforcing, but the mechanisms are a little different, as we'll go over in the next few slides. We can describe this type of behavior with the modified version of the Michaelis-Menten equation you see here, where n is the Hill coefficient, indicating the degree of cooperativity. If n is 1, we're left with the regular Michaelis-Menten equation that we're already familiar with. This is governed by Vmax and Km. If we increase the Hill coefficient to 2, then we start to see some sigmoid shape in the curve, and it becomes steepest right at the point of Km. If we increase the Hill coefficient to 4, then the curve becomes even more sigmoid and steep at the point of Km. What we're seeing here is the rate of enzyme catalysis V becoming more and more switch-like with respect to substrate concentration. We can go from a point of low activity to a point of very high enzyme activity just by slightly increasing the substrate concentration. We'll go over some concrete examples to illustrate how this works on the next couple slides. The first example I'm going to give is not really enzymatic, but it's the classic example of cooperativity, and that's oxygen binding to hemoglobin. This is actually how the Hill equation was first developed, to understand and mathematically model this physiologically important binding process. Remember from our lecture on amino acids that hemoglobin exists as a heterotetramer in red blood cells, with 2 alpha chain subunits and 2 beta chain subunits. Each of the 4 subunits has a heme group that binds oxygen, and when one of the 4 subunits has oxygen bound, this induces a conformational change that's propagated to the other subunits and makes them even more favorable for oxygen binding. So as you can see, the more oxygen molecules are bound, the more favorable the next binding event will be. This is what gives rise to the cooperative nature of oxygen binding by hemoglobin. We can describe this behavior with a modified Michaelis-Menten equation that has a Hill coefficient of around 3. This works out great for our physiology, because what it means is that in the high oxygen environment of our lungs, hemoglobin favors oxygen binding. But in the relatively low oxygen environment of all the other tissues in our body, hemoglobin favors oxygen release. So as you can see, this cooperative behavior is crucial for the ability of hemoglobin to provide all the tissues in our body with oxygen. This example isn't really enzymatic though, so let's go over another example on the next slide to show how that can work. I want to quickly point out that there's not really a standard mechanism for generating cooperative behavior in enzymes, but I'm going to show you one example here. It's between the protein kinase glycogen synthase kinase 3 and its substrate glycogen synthase. As an aside, it's pretty typical for enzymes to be named by their substrate and what they do to them. This is a nice naming convention because it makes it easier to keep track of everything. So we can see here that the kinase GSK3 phosphorylates glycogen synthase. One thing that we haven't talked about yet is that enzymes like kinases will often have short amino acid sequences on their target protein that they bind and modify, called substrate motifs. For GSK3, this is the amino acid sequence of its target protein that's bound at the active site and then phosphorylated. What's interesting about the substrate motif for GSK3 is that it's not just any peptide sequence. it actually recognizes and binds to a substrate motif that already includes a phosphorylated residue. You can see here the substrate motif for GSK3 is serine XXX and then phosphoserine. Those X's can be any amino acid, but GSK3 needs to have the serine and the phosphoserine at these positions in order to bind. The N-terminal serine residue that's bold and underlined is the one that'll be phosphorylated, but this won't happen until the other serine residue is phosphorylated first, so it can be recognized and bound by GSK3. In this figure below, I'm showing you the short stretch of sequence on glycogen synthase that gets phosphorylated by GSK3. And you can see there are all these serines all lined up in a row and ready to go. They need a priming phosphorylation to get started though. This is provided by another protein kinase, casein kinase 2. Once that happens, now we have our first GSK3 recognition sequence on the right. And then once that's phosphorylated, then we have our second GSK3 recognition sequence, and then our third, and so on and so forth. What's happening here is that the more glycogen synthase gets phosphorylated, the faster the rest of its residues will be phosphorylated. And that's how we get cooperative behavior in this system. Alright, so now we're going to end our discussion of enzyme regulation by covering different types of enzyme inhibitors. This is one of the largest classes of drugs. For example, I did my PhD looking at kinase inhibitors as potential anti-cancer therapies. When we think about different enzyme inhibitors, there are two main classes that I want you to consider, competitive and non-competitive. We'll go over competitive inhibitors first on this slide. These are going to be small molecules that bind to the active site and compete for binding with the substrate. What this means is that we'll alter the Michaelis-Menten Km, but not the Vmax. Let's look at an example here to demonstrate how that works. Here we have an enzyme that binds blue triangles and cuts them in half. A red triangle-shaped inhibitor can also bind to the active site and block substrate binding. But what happens when you increase substrate concentration to be much higher than the inhibitor concentration? They're both in binding equilibrium with the enzyme, having their own K-on and K-off rates. If you increase the amount of substrate present, this will swamp out the inhibitor, and you can still get up to the same Vmax for the enzyme, albeit at a higher substrate concentration, hence the change in Km. Let's switch now to talk about non-competitive inhibitors. Instead of binding at the active site and competing with substrate, these bind somewhere else on the protein that's distant to the active site. This is called an allosteric site. Let's walk through how that works here. We have our blue triangle binding enzyme again that binds and cleaves blue triangles, but now it has an allosteric site as well, and we can target this with a red square inhibitor. When the square inhibitor binds, it causes a conformational change in the active site that reduces the rate of enzyme catalysis. We can imagine a theoretically quote-unquote perfect non-competitive inhibitor that blocks catalysis but doesn't affect substrate binding. If this is the case, then only Vmax will be affected by adding in the inhibitor, but not Km. Let's look at how that works here. If we increase the amount of substrate, it can still potentially bind to the enzyme, but it can't be catalyzed as efficiently. That means that the Vmax will be lower, but the Km can stay the same. Of course, usually substrate binding will be affected too, so usually you'll have some kind of increase in the Km as well, but that's not technically required for non-competitive inhibitors. Alright, so we've gone over enzyme mechanisms, enzyme regulation, and enzyme inhibition. Now we're going to apply all of this to look at a potential drug target for COVID-19. And to explain how this works, we need to come back to this concept of polyproteins. Remember that viral genomes have this huge evolutionary pressure to remain compact, so they can fit inside of the virion. And one way viruses accomplish this is by compacting multiple proteins together into single-chain polyproteins. Each polyprotein is encoded in the viral genome by a single open reading frame, or ORF. This is how we refer to a continuous stretch of codons in the genome with no stop codons. When you think about it, if we have 3 out of the 64 codons coding for stop codons, then if the sequence was completely random, we can calculate using binomial probability that we'd expect to see a stop codon once every 20 codons. If we see longer stretches of nucleotides in a specific codon reading frame that don't have any stop codons, then it's very likely that these are protein coding genes. So that's what ORFs are. We see these all throughout our genome for every protein in our body. What's interesting in the SARS-CoV-2 genome is that 2 of its ORFs code for polyproteins. These are referred to as ORF1A and ORF1B. ORF1A codes for 2 different proteases that we'll talk more about on the next few slides, and ORF1B codes for 2 proteins that are critical for replication of the viral genome. The first one is RNA-dependent RNA polymerase, and the second one is a viral helicase enzyme that unwinds nucleotide double helices. The individual enzyme activities for each component of the SARS-CoV-2 polyproteins could, in theory, function just fine as part of the larger polyprotein. It could be like a big enzyme complex that has lots of different activities all together. But we know from experiments on related coronaviruses that they need to be processed into separate proteins before they're fully active, and this processing step is absolutely essential for the viral life cycle. Here we can see how this processing works. The two proteases from ORF1A are the ones that chop up the polyproteins into their individual functional components. You can see the protease cleavage sites marked by blue and red arrows here. Blue for the sites cleaved by papain-like protease, and red for the sites cleaved by 3C-like protease. This diagram is actually for the SARS-CoV-2 related coronavirus from the MERS outbreak. That's short for Middle East Respiratory Syndrome, which we saw during the 2013 coronavirus outbreak in the Middle East. We don't yet have as extensive a mapping for the protease cleavage sites on the SARS-CoV-2 viral polyproteins. We can make sequence-based predictions, but we don't have experimentally verified results yet. That said, it's a good assumption that the cleavage sites for SARS-CoV-2 are very similar to what we see here, because the two viruses are so closely related in sequence. And one thing we do know is that both of these protease activities are absolutely required for viral replication and spreading the disease. The 3CL protease, also known as the main protease MPRO, has emerged as the most promising drug target because there's no related human homologue, and therefore it's less likely to have off-target side effects. So how do we find a good 3CL protease inhibitor? I've written out the drug development process here. First, we need to develop an in vitro 3CL protease assay by synthesizing and purifying the protein, optimizing assay conditions, and coming up with a method to monitor the reaction. Remember that colorimetric or fluorescent assays are best because these scale well and allow us to screen large drug libraries and then follow up with more experiments on the hit compounds. We're fortunate in one way with the current pandemic, and that's that we've seen very closely related coronavirus outbreaks before, SARS in 2003 and MERS in 2013. Because of this, we have a head start on assay design, and we already have a good fluorescent assay for the 3CL/MPRO protease. This assay uses a phenomenon called fluorescence resonance energy transfer, or FRET, and the way this works is pretty simple. You have two fluorescent proteins linked together by a short peptide that contains the substrate motif for 3CL protease, that is, the sequence that it recognizes and cleaves. Remember, we can make this FRET reporter construct artificially as a single polypeptide from N-terminus to C-terminus, kind of like a viral polyprotein in fact. So the trick is this. When the two fluorescent proteins are linked, the FRET signal they give off is different than when the protease cleaves their linker peptide and they're separated. We can monitor the loss of FRET signal, as shown in figure C on the right, and then use this to determine the dp/dt enzymatic velocities. Once we have an assay like this that can be performed in a test tube, it's relatively straightforward to adapt the assay to high throughput format with liquid handling robots. This lets us rapidly screen large drug libraries and then follow up on the hits in more detail. The way we compare the binding strength of different drugs is with their IC50 values. IC50 is the inhibitory concentration at which the enzyme is reduced to 50% of its maximal activity. The way we find IC50 values is to repeat the assay with varying concentrations of inhibitor while keeping all the other conditions of the assay constant. This gives us a sigmoid plot like this. At low drug concentrations, we're at maximal enzyme activity, but as the drug concentration increases, enzyme activity falls. You can see this as we move from left to right on the IC50 plot. The way we calculate IC50 value is by identifying the drug concentration at which we observe half maximal enzyme activity. The tighter the drug binding, the lower the IC50 value, meaning that we can get the same effect using a lower dose of the drug. We always want the IC50 to be as low as possible, but not because we're trying to save money by using less drug. The reason we want low IC50s is because if we use the drug at lower concentrations, it'll be less likely to bind and inhibit other off-target enzymes in our body that we don't want to mess with. Off-target inhibition is a major source of drug side effects. So if we have a really tight binding drug that we can use at low concentrations, this helps reduce the risk of off-target side effects. After the first drug library hits are identified, we want to modify and improve these lead compounds to get even lower IC50s. One of the best ways to do this is by a method called structure-based drug design. In this method, we look at the protein structure of our target enzyme with the lead compound bound in the active site. And then we look around the active site and try to figure out what modifications we can make to the lead compound that would make it fit even better in the active site and have even better hydrophobic and electrostatic interactions. Using this information, we synthesize new variants of the drug, test them in vitro, and even determine the structure of some of these new compounds bound to the active site as well. Following this as an iterative process, we can improve the potency and selectivity of our drug candidate. In this case, the first crystal structure of the SARS-CoV-2 protease with a 670 nanomolar lead compound was published in April this year. And a follow-up study, published in June, identified a new compound that's now down to 40 nanomolar IC50, which is right in the range we want for actual drug candidates. Alright, so that's all for today. We talked about enzyme catalysis, regulation, and inhibition, and then we used all that information to look at the current state of COVID-19 drug development, including a deeper look at inhibitor IC50s. Thanks for watching, everyone, and I'll see you in class on Thursday.